Okay. This is for Bio 207. It's going to be the second part of the muscle chapter, kind of starting with your length-tension relationships and going into muscles, the different muscle types, and some of the different characteristics of the organ as a whole. All right, let's see. All right, so this is just a review at this point in our previous lessons and in Kelly's lessons. You should be comfortable now with the neuromuscular junction. Nope, that's not going to work. Here we go. The neuromuscular junction here, how you have a motor neuron communicating with the motor end plate on the sarcolemma of a muscle cell, releasing acetylcholine, interacting with the acetylcholine receptor, which leads to sodium ions being allowed from the outside of the cell into the cell, and that changes the electrical properties of the transmembrane potential, which causes an action potential. And so the action potential will excite and continue to propagate along the sarcolemma and down in the T-tubules. And that leads to the changes in the membrane of the organelle of the sarcoplasmic reticulum, which leads to opening of calcium channels, and calcium then can exit the sarcoplasmic reticulum and enter the sarcoplasm and bind to troponin on our actin, allowing actin and myosin to cross-bridge, and in the presence of ATP, a power stroke, a hinge occurs, and we end up with the sliding filament theory. And then for relaxation, we stop the acetylcholine triggering excitation of the membrane at the neuromuscular junction. We pump calcium back into the sarcoplasmic reticulum, so calcium levels fall. We don't have actin and myosin able to interact. And then with the help of factors such as the elastic properties of the connective tissue around the muscle, the elastic properties of the ligaments and the tendons, gravity to some extent, we get the return of the sarcolemma to resting conditions, and that leads to relaxation. Okay, so that was the first part of the chapter and the big three concepts of the neuromuscular junction, excitation, and contraction. So now we want to talk about, in a muscle, how and what happens when it does contract. What does that mean for tension development, for force production? And we have to start with a myograph experiment. So this is where we take some muscle out of the body, we remove the neuromuscular junction, but we mount the muscle on a device that allows us to electrically stimulate, so we mimic in some ways our neuromuscular junction, and we mimic that, causing excitation and contraction, and then we see what happens to the muscle under the influence of excitation and contraction. We generate tension, and it's called a twitch. And when we do these experiments on the myograph, we see a few things that we have to talk about. The first is the latent period. And the latent period is this time from when the electrical signal leaves from your hand, the button is pushed, to the point where you start to see a change in the muscle and development of tension. So it's typically a concentric, eccentric, or isometric contraction, tension developing. And this time where the stimulus to the start of that is the latent period, and it never changes. No matter if you make the force or the stimulus stronger, no matter what happens, this is the period of time that is standard, where there's no change in the muscle for this short period of time yet. So it represents the excitation of the membrane, the sarcolemma, the movement of the action potential and excitation into the T-tubule, the release of the sarcoplasmic reticulum's calcium, and the calcium going and binding to troponin. So it represents that time before we get cross bridges and we get power strokes and pivoting of the myosin and sliding and contraction and tension building at the sarcomere level. So the contraction phase would be the part of the twitch where we see increases in tension, and so we would expect to see increases in calcium availability increases in ATP being utilized and increases in cross-bridging of actin and myosin, and we see the area of overlap increases, the I bands go away. Then over here in the relaxation, we would expect to see that there's no longer any excitation on the membrane, so we start to now pump calcium back into the, that's supposed to be calcium. We start to pump calcium back into the SCR, so calcium levels fall. As calcium falls, there's less troponin-calcium interaction, so now tropomyosin can cover the actin binding site, and so now we have less cross-bridging of actin and myosin. So with less cross-bridging, we have less tension being able to develop, and so we start to see relaxation occur. Okay. Now, not every muscle cell is the same, and what this graph is trying to show you is that we have different cells in the body, depending on location, that will generate under the same stimulus. They generate a different twitch profile, meaning they have a different time for contraction and relaxation, but they all meet about the same amount of tension. And this is where we get things called fast twitch, slow twitch. So the twitch of your eye muscles, which is constantly changing the eyeball's focus, so your little 3-degree window of light penetrating the eye and focusing upon your fovea centralis is constantly being barely little minute adjustments, so it's extremely fast twitch. Your gastrocnemius and your soleus muscle are the calf muscles, and we tend to use them as great examples because there is a nice division in the two muscle groups, the gastroc being a fast twitch muscle, so a white muscle, and your soleus being a good representation of a slow twitch muscle, a red muscle that, you know, does posture and maintains the ability for you to walk, whereas the gastroc helps you jump. All right, and so these are basically the twitch profile. When we compare cells from different muscle groups, might be able to tell us if the muscle cell has a specific twitch profile or not. So fast twitch versus slow twitch or intermediate twitch. Now, that's one area we can look at different cells from different areas. When we focus upon a specific cell, one of the questions asked for skeletal muscle is, how can I make the twitch, the amount of tension it produces, go up? But I can't really change the amplitude of the stimulus, meaning I can't change the voltage applied. I can only change the frequency that I apply a set voltage. And when I look at changes in frequency, changes in the stimulation rate on a single muscle fiber, as I make the rate or frequency of stimulation increase, I will actually see increases in the maximal amount of force that I can generate. All right, and so we have different names for the changes in frequency on a single muscle based upon what the myograph starts to look like, what the twitch profile starts to look like. And so the first one, wave summation, is basically when we start to increase the twitch frequency such that before the muscle completely relaxes, we start to a second stimulation. And so before we get complete removal of calcium, before we get complete removal of actin and myosin interactions, we stimulate the next excitation event, so we get more calcium and potentially the sarcomere is in a more optimal position for length-tension relationship, and so we can get a stair-step increase in the tension we produce for the set muscle cell. Okay, and if we continue to do this and we start to reach a max, it's called incomplete or unfused tetany. And so again, you will get a plateau in some sense, but the plateau still shows rise and falls, hills and valleys at a tension that's maxed out. But again, incomplete tetany, you're going to see the frequency rate still allows some small little bit of relaxation, but it begins to hold at almost a max tension rate. It keeps hitting that max tension rate as you continue to apply a stimulus at that frequency. All right, now this amount of tension developed in incomplete or unfused tetany is still not the maximal amount of tension a muscle cell can generate. You will get the maximal amount of force, all right, in a frequency rate that never lets the muscle cell enter any state of relaxation. And when you do that, it's called fused tetany, or it can be called, from your book, complete tetany. And this is characterized by, it reaches a maximal tension development, it reaches a plateau, it reaches a steady state, and you never see any drops in tension. You never see any drops in tension because the frequency rate is so fast that you never get any state of relaxation for that individual muscle cell. Okay? And so if we go through the pictures of your book, here is what's called TREP, where I increase the tension developed by an individual muscle cell by increasing the frequency of stimulation. But in this scenario, I'm letting the muscle completely relax, so it completely contracts, completely relax, and so I'm hitting it with the next stimulus at the second it completely relaxes. So you're running the possibility that there still may be some calcium ions and maybe a few cross bridges, and you haven't quite returned to steady state, full relaxation on the sarcomere position. And so you are making a little bit more tension because of those stray calcium, maybe a few stray actin and myosin cross bridges, and again, because you haven't quite put the sarcomere back to its original position. Wave summation, you start to, before complete relaxation occurs, so in the middle of the relaxation state, you hit it with the next stimulus, and you'll get a stair step. And if you continue to do that, you'll end up in what's known as incomplete tetany. So you'll wave summate to a point, and then you hit a tension line, all right, a maximal amount of tension, but you're going to continue, you're not steady state. You continue to see, because the frequency is coming with some relaxation, you continue to see hills and valleys. And then complete tetany, you get a maximal amount of tension, but you get true steady state, basically a straight line where you're holding that tension ability because you never let the muscle relax. Okay? Now, that's at a single cell level. Once we convert and start talking about a muscle grouping, remember that we're talking now a motor neuron and all of the muscle fibers it controls, which is a motor unit. And in that motor unit, so in this example, our little orange guy here controls 6 muscle fibers. And when that motor unit gets stimulated, this motor neuron carries an action potential down, and as we talked about previously, the synaptic knobs are going to talk to each individual muscle fiber at the sarcolemma, going to change, release acetylcholine, change the excitation of the sarcolemma, and lead to these muscle fibers to contract. And so those 6 are going to give us this twitch profile for this muscle. If that is enough to lift a piece of paper, then that's all that happens. But let's say we're trying to lift a heavy book. If this motor unit can't lift and make the force, then we might have to recruit another motor unit. And if it's still not enough force, we recruit a third motor unit. All right. Now, one thing to note is typically when we talk about a motor unit, let's say the orange here is a motor unit for all slow twitch fibers. So this is going to be a motor unit that controls 6 muscle fibers that are all slow twitch. Versus, let's say our purple is a fast twitch motor unit. So it's going to control 6 purple muscle fibers that are considered fast twitch. So typically these motor units tend to control all the same type of muscle cells. Okay? And it's key in that while the tension produced at a cell level can increase with increasing frequency, the whole muscle, that will play a role in getting more tension to develop in addition to recruitment of more muscle fibers. And when you get a maximum amount of contraction, you get every single muscle fiber, every single motor unit involved in that muscle contraction. Now, these are just some slides to go through a few different properties of how things work at the organ level. So load and speed of contraction, they're inversely related. What that means is if you have a small load, your distance shortened is the biggest, and you can do it in a very quick period of time. okay? Versus if it's heavier the load or the heavier the resistance, it takes longer to shorten. It takes longer for that shortening to begin, and the muscle will shorten less, right? And then you'll recruit, and you'll probably have to get more muscle cells involved through recruitment to maybe accomplish that load. All right, now moving on to energy. ATP is your energy molecule in your muscle, and so your ability for the muscle to continue to contract, continue to produce tension, to continue to overcome the force is in many cases dependent upon how much ATP is present. Now, your muscles have a little bit of ATP inside to be able to start a contraction. So as long as you're doing something that takes 2 seconds or less, you really don't make ATP to do that. You just use the ATP you have already present in your cells. As you go into something that takes 10 seconds, 15 seconds, you're going to have to start making a little bit more ATP. And the easiest way to do that, because again, you're only taking 16 breaths a minute, so you might not be able to gin up aerobic or even anaerobic means, because glycolysis is an 8-step process. You have this mechanism of creatine, all right, has a phosphate, and you can take that phosphate, bind it to a adenosine diphosphate molecule, and get ATP really quickly. You can also do, with the enzyme present, ADP plus, there's an ADP, right, and that'll give you, so this one is going to donate a phosphate over here, and so you'll end up with an ATP plus an AMP, all right, which is an adenosine monophosphate. So these kind of mechanisms here, all right, allow you to make more ATP without having to really burn any fuel. Right? You just need a few enzymes and maybe some phosphate stored in some form within the cell. Once you get into longer activities, so normally it's 130 seconds or more, you're going to have to start breaking down fuel to maintain ATP production to meet the demand required to make all those sarcomeres and actin and myosin cross bridges interact and slide. All right, and so this is where your breakdown of glucose is going to generate ATP through the glycolysis, and then the pyruvate can enter the mitochondria, continue to be broken down, as well as fat, your background levels of fat being broken down by the mitochondria to give you your resting rate of ATP will gin up as well. Okay, so when we look at this, for any short-term mechanism, and again looking at the table, if something needs to happen less than 17 seconds, we only really need this ATP that's already there and a few creatine phosphates, and maybe even that ADP transition to ATP and AMP with 2 ADP molecules. And that gives us enough energy to do something very, very quickly. All right? If we're going to go beyond that 15 to 17, 20-second window, we're going to have to start utilizing the breakdown of fuel. So we'll utilize glucose breakdown through glycolysis, and then glycogen broken down into glucose, which then can be broken down through glycolysis to continue to give us energy. All right? Now this is going to enhance energy that's being made by fat breakdown. All right, you always have some background fat being broken down by the muscles, but remember, you're trying to meet a load. So your demand for ATP is over what the background levels of fat being broken down can make in ATP production. So what this is trying to show you is a muscle cell is maintaining tone. It maintains some contraction. It maintains some level of ATP always available. And it does that through breaking down fatty acids. And we get fatty acids from triglycerides in the blood. We get fatty acids. Some of them can be found in HDL and LDL cholesterol molecules because it helps, cholesterol travel in the blood. And they come in and they get broken into 2 carbon molecules that's supposed to be a C, and those 2 carbon molecules enter in the mitochondria, and if this is let's say an 18 carbon fatty acid, that gives us 9 2 carbon molecules, so I'm making 9 times the ATP for this molecule, whereas glucose is only 6 carbons. So this is all bioenergetics. And so at rest, I use fatty acids, I make my ATP, I have my background ATP, and for glucose, I just spare it, and I make glucose into stored glycogen. Now, I can only store so much glycogen in the muscle, so any glucose that is not going to be stored as glycogen and not going to be used for energy, the muscle will not take it in, and it'll actually go to fat, and that's where it gets converted into fat. Okay? Now, when I start moderate activity, so low intensity, I increase my ATP demand to make forces, all right? And I'm always going to have some background fatty acids. You know, they might only make, let's say I need 150 millimoles of ATP to do my activity, and my fat makes 100 out of the 150 needed. So my fat is going to continue to be burned, but to compensate for that extra 50 millimoles of ATP, I'm going to need glucose to be broken down to pyruvate, and that's going to generate some ATP. And then some of that pyruvate will enter into the mitochondria and the Krebs cycle and the citric acid cycle and the oxidative phosphorylation and make me some more ATP. And so through this process, I make my extra 50 millimoles of ATP on top of my 100 from my fat, and I'm able to meet my moderate demand of exercise or activity. Okay? And this, we see glycogen starting to be broken down. And so for most of your like long distance, long activities, you continue to break down and use a combination of fat and sugar to meet your energy demands. Okay? Now, when you go really, and this is really high intensity, all right, very high, all right, you start to, again, you're still burning fat, but you are really having to overcome the deficits that fat can't make in ATP production, and it takes a while with breaking down glucose into pyruvate. But in order to keep this going, right, part of what you have energetically in the process of glucose becoming in pyruvate is you're making NAD into NAD, and this is H, okay? And this NADH has to be made back to NAD to keep glycolysis running. So what happens is to keep glycolysis running, I got to get rid of this H. And that's where the H will be dropped. Pyruvate becomes lactate. It's lactic acid because the H is donated to the sarcoplasm and to the blood. And in doing that, I get back NADH to NAD. So I can make more glucose to pyruvate, make more ATP here, but in doing so, I generate lactate and hydrogen ions, so lactic acid. All right. Now, at some point, my pH, all right, is going to drop as my hydrogen ion concentration increases. And what ends up happening is as pH goes down and hydrogen ion content comes up, you start to mess with enzymes. And so you mess with enzymes and they no longer can make glucose become pyruvate. They can no longer convert ADP and creatine phosphate into ATP. And so all of a sudden you no longer have ATP available to meet the demand, and that's when you hit what's known as the anaerobic threshold, the wall, and you kind of crash. Okay. Now, at some point, you got to do something with this hydrogen and this lactate to get it back to pyruvate, all right, and to get NADs back to NADHs, and that's where oxygen debt comes into play. And so when someone stops activity, you know, that doesn't mean they stop and return to normal breathing right away. If they've accumulated lactate and hydrogen, they need to recover it back to a usable form of pyruvate and glucose and NADH. And so that's where oxygen debt comes into play to help them convert all of these back. And that's known as the Cori cycle. Okay? So, muscle fatigue. When muscles no longer perform their function, they are considered fatigue. Right? And you can have muscle fatigue because of metabolic reserve. You run out of glucose to make the ATP to pyruvate, you know, so you run out basically of fuel for making ATP. No ATP, no cross bridges, no sarcomere sliding. If you mess with the sarcolemma or the sarcoplasm, you're working at the acetylcholine, all right, or the calcium level. And if you mess with acetylcholine or calcium, you mess with the neuromuscular junction, and you mess with, that's supposed to be any excitation, and so that then will lead to fatigue, because if you mess with acetylcholine and calcium, you can't get contraction, even if you have lots of ATP. Lactic acid, low pH, any changes to pH mess with all of your pumps, all of your enzymes, all of your proteins, and they denature or they get no longer as effective or efficient with kinetics. And so you will have problems making ATP or pumping calcium or moving calcium, and so that will affect your, again, sarcomeres, changing length and generating tension. And muscle fatigues result in pain and exhaustion because you run out of ATP, you have oxygen debt, and then if you change pH and you change intracellular reserves of things, you are going to see that your fatigue, causes neurons on the sensory side to be triggered for pain or temperature and itch. Okay? So you can have fatigue if you change or damage the ability to make ATP, you change the sarcolemma and its ability to interact with acetylcholine, if you change calcium, and in a sense, fatigue is going to damage and eliminate these three molecules critical to causing an action potential from working as effectively anyway. Okay. Again, going back to lactic acid removal, at some point you have to get the lactic back to a usable form, and so lactate can travel to the liver, become pyruvate in the presence of oxygen. So oxygen has to be here, all right? And then pyruvate through gluconeogenesis in the liver can become glucose and then return to the bloodstream, and it can go to the muscle, it can go to the brain, it can go to other organs and be used for energy, all right? And this is called the Cori cycle. In the muscle, a small percentage of lactate can become pyruvate again, but this only happens for a very small portion of the lactate. The majority of it has to go to the liver. And you can't get pyruvate back to glucose. So not a lot of it becomes pyruvate in the muscle because, again, then you end up with a problem of too much pyruvate, and maybe you don't want to run all those mitochondria and burn all that pyruvate. All right, and so oxygen debt, again, because you have to have oxygen to make lactate become pyruvate again, that's part of the reason why you continue to breathe heavily and pant at the end of exercise. Now, depending on your fitness level, will determine how much lactate you have and how quickly you can convert it back to pyruvate and a bunch of factors. And so we can look at how much time you spend in oxygen debt and then basically make assumptions about your fitness level. Okay. The last little bit of the chapter is, okay, we have muscle, we now know how it contracts. How are other ways we can remodel, reconfigure, or influence the muscle's ability to either increase in size, which is anabolics, all right, or decrease or burn or make more energy, so catabolic reactions. Remember, metabolism is defined as all the chemical reactions. And so these are some of the big four hormones. Growth hormone is going to help us grow more muscle, so it's actually going to help anabolically, building more muscle, making more protein, making more actin, more myosin, more sarcomeres, will hypertrophy the muscle. Testosterone, same thing. It helps us increase muscle mass. It helps us increase actin, myosin, sarcomeres, myofibrils. So these are typically going to influence the muscle through anabolic needs. Okay? And so if you look at your baseball players or your athletes, if they want to increase their muscle mass, they tend to abuse androgens, which are testosterone precursors, and growth hormone. These two are going to work more at the catabolic level, all right? So the breakdown, and they specifically usually work at the energy consumption level for ATP. So typically if someone has high thyroid levels, they tend to be shaky, they have low body fat, they burn a lot of fat, their muscles are constantly kind of twitching and working because they're constantly making lots of ATP. And then epinephrine, think of it as adrenaline, fight or flight. So high levels of epinephrine, you either are running away or fighting, so you would expect ATP production to be high, glucose levels to be utilized for energy expenditure. So those are some of the effects of the hormones. And this table kind of summarizes some of that. So again, epinephrine, you're going to basically try to run away. So you would want to break down glycogen and use glucose for energy. Norepinephrine, which usually accompanies epinephrine, typically works not on the muscle itself, but on other things to help make that fight or flight response. Growth hormone, again, you stimulate growth. You stimulate protein synthesis, making the muscle bigger, right? Testosterone is going to, again, protein synthesis, make more actin myosin, more myofibrils. Estrogen doesn't work as well as testosterone, but for some, it will have some effect on skeletal muscle and some changes to glucose utilization, all right, and not necessarily as much on protein synthesis. Cortisol, our stress hormone, is going to work on skeletal muscles. It's going to make the skeletal muscle utilize and build more protein and utilize more fat for energy and try to spare gluconeogenesis, because it's a hormone that's trying to say you're stressed out, you got lots going on, let's keep the glucose for the brain and not really use it for energy consumption right now. You might need it later. And then insulin-like growth factor results from growth hormone, and it basically, like growth hormone and testosterone, stimulates protein synthesis, which stimulates increases in muscle mass. The last part of this chapter is going back and really looking at fast twitch, slow twitch, and intermediate fibers. There now, if you look at exercise literature, there are all types. You have intermediate fibers, multitude of categories. But for our book, we're going to try to keep all of those intermediate fibers just kind of one big group, and then we're going to use these as our two extremes of fast twitch versus slow twitch, and then intermediates in the middle. Right? And I throw this picture on here because basically, depending upon what kind of fiber you have, it might stain in a muscle cross-section different colors because of the intracellular components within that fiber. So our red fibers tend to be our slow twitch fibers, and they're red because they have lots of oxygen available, because they have lots of protein called myoglobin. Like hemoglobin in the blood, myoglobin binds oxygen, so they can hoard oxygen in the cell. So they can use more oxygen for aerobic purposes. They have more mitochondria. They have more blood vessels around them. So you see that as red, and if we were to highlight this and look deeper, we would probably find more little capillaries in and around these muscles because they're bringing more oxygen to them. Versus the white over here and over here, they still have a little color, so they have some myoglobin, they have some mitochondria, but they tend to utilize more anaerobic means, so they're not going to have as much myoglobin or as much mitochondria or as many capillaries around them, so they're not going to stain as red. And then you can see there's lots of shades in between. So if this is our light and this is our dark, here's an intermediate, here's an intermediate, here's an intermediate, and there's lots of, and then there's these intermediates, so there's lots of in-betweens. Okay, and this is a type of intermediate. All right, so lots of intermediates. Okay, and so typically fast fibers are going to be contract quickly. So think about the eye, think about the gastrocnemius. They have a large diameter, large glycogen, few mitochondria. So they are typically going to use glucose, all right, and glycolysis, the breakdown of glucose to make ATP. They tend to have high creatine levels, and they are going to help you jump and run and do things, again, for that like 130 to 40-minute window. Okay. Now, they have some mitochondria. They have some ability to burn fat, but they have all the machinery up and running to do glucose and glycolysis and anaerobic needs. Okay? Your slow fibers, your red fibers, are going to be the opposite spectrum. So if The fast twitch were little mitochondria big. These are small diameter and massive amounts of mitochondria. They have a high oxygen supply. They have a lot of myoglobin, which, when bound to oxygen, forms a red pigment. In addition, they have high levels of capillaries. So with a lot of blood carrying oxygen into this muscle tissue and a lot of myoglobin binding oxygen, they get a very red appearance. Okay? And so if we look at body types, if we were to take cross-sections of Usain Bolt, who's our 100-meter, 200-meter sprinting god, we would expect a quadricep muscle and maybe most of his muscles involved in running to stain something like this. Does he have red fibers? Yes, but he predominantly has white fibers. Versus Meb, the marathon guy, or the British guy that won the 10,000 in the last Olympics, we would expect their muscles to look more like this with lots of red. All right, and if we were to probably look at these would probably be more intermediate fibers and maybe less true fast twitch fibers. Okay, and then most of us probably fall into this category. We have a good mix. We actually have lots of intermediate fibers, and whether we tend to be more of a sprinter or a distance runner, okay, depends upon our training and what we stress the body and stress these intermediate fibers to become more like. Okay, so from your book, here's what it says about intermediate fibers, and then it gives you this table, all right? And it uses these, or I gave you this table, sorry. And then it tells you typically you have type 1 fibers, which are your true slow twitch, the red fibers, and then you have your true fast twitch fibers, which are these type 2B fibers. And then all of your intermediate fibers tend to be in the middle. And as I'm showing you here, there's lots of different categories. If you really do get into this with exercise physiology, you're going to see there's even more categories of type 2X, type 2N, type 2M, type 2B, type 2A, you know, so there's lots of letters. The key is if this is one extreme and this is one extreme, these type 2 fibers are in the middle and they're going to have characteristics similar to the fast twitch and similar to the slow twitch, but they're a mix of the two. Okay? And this is important. I think Kelly is going to ask you 1 or 2 questions about this. So do look over these tables and be able to compare and contrast slow twitch versus intermediate versus a fast twitch. Okay? And this is, again, from your book. So I would say spend some time looking at this. All right? So diameter, small. In the middle, large. Peak tension, it takes a while. Really quick. In the middle. Contraction speed, slow, fast, fast. Now this can be trained to be more slow if you train it, okay? Fatigue resistant, very low because once ATP is gone through anaerobic means, it's done. Versus fatigue, very fatigue resistant over here because they have all those mitochondria and all that oxygen down to the myoglobin so they can kind of gin up and go and make lots of ATP for a long period of time. Red, white, and this is somewhere in the middle. Tends to be pink. Lots of myoglobin, some. Now again, you can make this increase in myoglobin with training. Capillary supply, you might have 4, 5, or 6 capillaries, whereas you might have 2 over here. And then this again will change depending on training. All right? Lots of mitochondria, a few, and then again, this will change depending on training, all right? How much glycogen you have stored, because you're going to use a lot of fat here, you're not going to store a lot of glycogen. And then part of glycogen content is dependent upon training, okay? So just review this table. All right, and then this is just more information if we look at where the muscles, most of our muscles and most of us are going to be good mixes. There are some muscles that tend like the soleus to be more red and the gastroc tends to be more white. But the gastroc actually does have two distinctive red little patches on it if you look at rat or mice or even us, if you look at it at a microscopic level, right? But most of ours tend to be mixed with maybe based on genetics, based on training, we tend to maybe shift towards one or the other, okay? All right, muscle hypertrophy. When our muscles grow, sometimes, depending on injury level, we can get myosatellite cells to join and make a new muscle fiber or fix an old muscle fiber. But most of the time what training does is it takes the muscle fiber we have and pushes it to increase sarcomeres, increase actin and myosin, increase protein synthesis, increase glycogen reserves, and the muscle gets bigger. And so this is what training does. Now, remember, all of these changes can take 6, 8, or even 10 weeks. So when you go to the gym, you know, it's about to be New Year's, people go to the gym, most of the changes you see in a muscle in the short term are basically going to be changes that occur with energy expenditure and things like that, neuromuscular muscle interactions, and not actually hypertrophy, right? So hypertrophy is going to be something that comes with extensive training and extensive loading and long periods of time. And unfortunately, muscle atrophy, lack of muscle, and therefore the loss of protein synthesis, the loss of proteins, the muscle decreasing in size can happen in as quickly as like 3 days. So you know how they say it takes 21 days to make something and 3 days to lose it? Basically, that's what happens here, all right? Or is it 21 days to break a habit? Anyway. Muscle atrophy, short term, it can happen. Hypertrophy, long period of time. This is why you see your muscle bodybuilders, they might have to take years to be at a competitive level of training, and then you get one little injury and they lose muscle mass very, very quickly. Okay? All right, well that is the chapter, so hopefully that helps prepare you for quiz 3 in Kelly's class, and in my classes, your test, which is October 31st. All right, stop recording.